Hi, and welcome back to today's lecture on antiviral drugs. This is chapter 8 in your textbook. It's a really boring chapter. So if you can't make it all the way through the chapter, I feel your pain. It took me a few tries as well. I have a helper here with me today who's going to hang out for the first few slides. Do you want to introduce yourself?
Yes. My name is Cora Nichols. This is my mom.
How old are you?
I am 9 years old and I am in 4th grade.
Yes, so Cora is going to help me kind of talk through some questions like I would with you guys to make things a bit more interesting. So if you're paying any attention to some of the latest news about the current coronavirus outbreak, you may have seen something about a malaria drug that seems to be somewhat effective in treatment of COVID-19. So down here in the slideshow is a link to that article, to the article in case you feel like reading it. What it seems to be showing, and this was from a group out of France, was that when people are treated with this drug, hydroxychloroquine, they have a decrease in PCR positive samples for coronavirus over time, whereas in our control, those numbers don't change very much. This is a really small sample. It's not a full clinical trial yet, but it looks kind of promising. And this is what the FDA is now being charged to study and put a lot of work into. So we'll see if this bears true in a larger study set. Okay, so I have a few questions that Cora and I are going to kind of talk through. And I want you guys to think about these things on your own. These are important questions to ask yourself. So the first, Cora, why, when you sometimes have a cold or a sinus infection and your doctor tells you it's caused by a virus, why do you think you're told to just go home and wait it out and you're not given a prescription for anything?
Well, you should go home and wait it because the virus will eventually run down and run out of power.
Mm-hmm. So
So
most infections with viruses are self-limiting, and after a while, your body will clear the virus and you won't have to worry about it anymore. Another reason is because we actually don't have a lot of good drugs that treat viruses.
that's sad.
That is sad.
Yeah.
So I want you guys, you students listening, to think about what you remember about our antibiotics, about those we use to treat bacteria. So knowing what you do about the number of antibiotic drugs, about how they work, why do you think it's unreasonable, Cora, for people to simply say, well, just make more drugs?
Well, you don't really have a lot of materials or supplies to make the drugs, and if you do, it takes a long time.
That's right. It does take a really long time. We'll talk about that in a couple of slides. The process of just making a new drug is not so simple, especially at a time like right now when there's a new virus we're trying to deal with. We can't say, make a new drug and have one show up like next week. That's not how drug discovery works. So now thinking about what you already know about viruses from listening to the last couple of lectures or from having a mom who's a virologist, Cora.
Yes, that's how I know everything.
Why do you suppose we have less antivirals on the market, less drugs to treat viruses than drugs to treat antibiotics? What do you think, Cora?
Because viruses are more different. Like the variety of viruses is larger,
Mm-hmm.
and the virus,
Yeah.
the like, bacteria
Mm-hmm.
is very similar. Like
Mhm.
all bacteria are similar to each other.
Mm-hmm.
I just forgot a word there.
That's okay. You're right, though. Bacteria all have certain things in common.
Variety. That's
Variety, yeah. So bacteria all have certain things in common, like they all have similar cell walls and similar ribosomes. But viruses, even though there are viruses, they're all really different even from each other. So a drug that might work against one kind of virus doesn't work against another kind of virus because those two viruses have different structures. Yeah, exactly. All right, so today in our lecture, these are our learning outcomes, the things that you guys are going to know. I want you to be able to identify the main targets of antivirals and how they're informed by the concept of selective toxicity. That is, how do they inhibit viral replication without hurting our cells or our bodies? To explain a few specific drugs that are mentioned in these slides. So you only need to know the activity of oseltamivir, also called Tamiflu, which some of you may have taken before. Maybe you took it this year if you were diagnosed with the flu.
My friend actually took that.
Oh, you had a friend take Tamiflu this year. Yeah, it's commonly prescribed. Amantadine, acyclovir, ribavirin, and AZT. So these are the only ones that you need to know specifically how they work. And then the last thing is to understand the basic concepts of the development of antiviral resistance, which is not as complicated as antibacterial or antibiotic resistance in bacteria. Okay, so normally I would talk you guys through a replication cycle and have you work in groups to think about where during the replication cycle you would design a drug to inhibit replication. So instead, Cora is going to do that with me today. So what we have here is the replication cycle of coronavirus. Usually I talk about Ebola because Ebola is really interesting, but you know, with the times we're in right now, I thought we would do coronavirus.
Yeah.
So step one is attachment. So this is for SARS, which is closely related to the coronavirus we're dealing with right now. So it attaches to a cellular receptor. That's step one. And this triggers receptor-mediated endocytosis. So that's step two, penetration. So our virus will be in an endocytic vesicle. And the membrane, because coronaviruses do have an envelope, will fuse with the endosomal membrane, releasing the genome into the cytoplasm. So this is step three, uncoding. We're releasing the genome into the cytoplasm. Now, all of this stuff right here is step four, biosynthesis. So I'm just going to talk you through a few highlights of this. Recall that coronaviruses are positive stranded RNA viruses. So this positive strand of RNA looks like a messenger RNA. And it can, a portion of it gets immediately translated into proteins. So it makes these two large polyproteins, meaning many proteins linked together, PP1A and PP1AB. Now these huge polyproteins then get cleaved, they undergo proteolysis by various proteases into smaller proteins, and these smaller proteins make up the polymerase, so the viral polymerase. This viral polymerase now interacts with that positive strand RNA genome, and it's going to make some, up here we have our negative strand RNA, and then that negative strand RNA gets used to make all of our messenger RNAs. Those various messenger RNAs are then translated into the viral proteins. So this happens here in the endoplasmic reticulum. So all of this stuff, making new genomes, making that negative strand RNA and messenger RNAs, and making proteins, all of that is step four of biosynthesis. That's a lot of stuff, right?
Yeah.
Yeah. So once we have our new genome copies and our new viral proteins, they go through step five, assembly. So they assemble in the ER, they bud through the ER, so their envelope actually comes from the endoplasmic reticulum. They bud through the ER, and they are released in step six, released into the extracellular space by the exocytosis pathways of the cell. All right, so we've got attachment, penetration, uncoding, biosynthesis, assembly, and release. Cora, if you were in the job of designing antiviral drugs, which of those steps would you target?
I would target penetration.
So this step here where we go from attached to inside, why would you target that one?
So that it can't make any more and it can't make you sick, so that it just gets rid of the rest.
So you say, let's just keep the virus from getting in the cell at all.
Yeah.
Right? Because if you stop this step, none of the other stuff can happen. No biosynthesis, no assembly, no release.
Nope.
Right. So I think that's a good approach, right? Stop it before it gets started. So let's say you couldn't prevent this from happening. You couldn't make a drug from this. What other thing might you try to prevent?
Well, then, I would try to prevent it from making more, I would try to prevent it from biosynthesis.
Okay, so maybe you would say, let's keep it from making more copies of its genome,
Yeah.
or let's see if we can stop it from making more of its proteins.
Yeah.
Yeah, that's a good approach too. That in particular, targeting biosynthesis, is what a lot of antiviral drugs do. They try to stop protein synthesis, or they try to stop replication of the viral genome. Yeah, there are others that work up here at the early steps. So there are some drugs that try to prevent attachment, some that try to prevent penetration or uncoating. And there's even some that work way out here at release and say, well, maybe we can't prevent these earlier things from happening, but maybe we can keep these new viruses from getting out to infect a new cell. Yep. All right. Thank you, Cora.
Okay.
For helping. I'll see you when I'm done.
Okay.
Okay?
Goodbye.
Bye. All right, guys. So let's move on and let's talk about some of these antiviral drugs. One of the issues with the treatment of viral infections is that we have very, very few antivirals in the market today. So there's only about 100 antiviral drugs that we can use in the United States. And it took a while before we really started developing them in earnest. So in our timeline here, we can see we discovered some viruses early on. There weren't really many drugs approved for their use. And then here in the 80s and 90s, we started kind of having a boom in the development of antiviral drugs. So a lot of it came after the discovery of HIV, and once we started figuring out how that worked, and we immediately, of course, started developing drugs against HIV, the same with hepatitis C. And if we look in 2016 at where most of these antiviral drugs, what they're targeting, 41 of those 100 drugs target HIV. And they target a lot of different parts of the viral replication cycle. About 18 target hepatitis C. We've got about 10 that will target the herpes viruses, and 8 that are specific for influenza. And a lot of these drugs don't work on different types of viruses. So a lot of these HIV drugs, they only work on HIV. Same for herpes virus drugs, same for influenza drugs. So when we think about things like spectrum, our antiviral drugs are very, very narrow spectrum. In fact, they often work just against one particular type of virus. Okay, so why are there so few antiviral drugs? And this is a question we hear a lot with antibiotics too, like why aren't there more of them? One of the issues with antivirals is that they can have some pretty detrimental side effects. So that toxicity level can be pretty high for some of these drugs. Also because they have to get inside of cells, they can have effects on the cells. Others need to be processed by the liver into an active form and can thus have side effects of liver toxicity or toxicity to red blood cells that results in anemia or other types of side effects. It can be really hard to make good antivirals for some viruses because of the use of host machinery. We can't target ribosomes when we talk about viruses because all viruses use our ribosomes. Some viruses use our polymerases, so we can't target polymerases without affecting our own cells. So the selective toxicity here can be pretty tricky depending on the type of virus we're talking about. And another issue is that there are some viruses that we can't grow in vitro. We don't have a good cell model for them, so we don't have a good way to test antivirals and see if they're going to work. Other viruses don't have an animal model, so we can't study how toxic this drug is going to be very easily. And then others are just too dangerous to work on. There's only a few places we can work on them. So moving into developing antivirals for those is very slow because there's not a lot of people working on it. So for instance, human papillomavirus, there's not really a good animal model for it, so it's hard to study the efficacy of HPV drugs in vivo, although we can kind of look at them in vitro. Smallpox, there's only 2 places in the US where you can work on smallpox, so we're not going to put a lot of people working on antiviral drugs for something like smallpox. And the same for some of our BSL-3, BSL-4 agents like Ebola virus and Lassa fever virus. Again, there's very select places in the United States and worldwide that can work on these viruses. So moving toward an effective antiviral for these types is just very, very slow. Now, like Cora and I were talking about earlier, where might you develop an antiviral drug to target replication? And we've done a pretty good job, if we look at all of our different compounds together, at targeting the different parts of viral replication. So we do have some drugs that prevent attachment. So these can be peptide analogs of the attachment protein. So they sort of like bind to and suck up all of the receptors on the cells, or they bind to the virus itself and prevent it attaching. So these are mostly for HIV. We can target penetration and uncoating. Again, HIV, some of the drugs against herpes simplex viruses do this. Targeting biosynthesis in the form of mRNA synthesis. Interferon works really well at this. You guys know interferon is the antiviral response of the body. We can give people interferon and it will decrease viral replication. So this is shown to be pretty effective against our hepatitis viruses, works pretty well against papillomaviruses. Or we can use antisense oligonucleotides. They would work like an siRNA, and they would bind viral mRNAs and either cause their degradation or prevent their translation. So we can use those against some viruses as well. Protein synthesis, initiation. Again, protein synthesis is kind of tricky. We can't use drugs that directly target ribosomes because then you're going to prevent your cells from making proteins as well. But we can again use interferon, which as the antiviral response does shut down protein synthesis. Only this is global protein synthesis as well. And we can try to prevent replication of the viral genome. We're going to talk about some of these today, some nucleoside analogs, and there are also non-nucleoside analogs. This is mostly, we'll talk about these when we talk about HIV, but we'll talk about nucleoside analogs today. So herpes viruses, HIV, hepatitis viruses, we have drugs that will target all of these. And then the last one is assembly and release. And so these are some peptidomimetics. They look like various peptides. And so these are against HIV and herpes simplex virus. So we can target various stages of the viral replication cycle, but it depends in part on what type of virus we're actually targeting. So the most important reason that it's challenging to develop good antiviral agents is that an antivirus must completely block viral replication. We can't use a drug that only partially inhibits replication. That would be completely unacceptable. And the question of why? Well, think about if you are only knocking down 80% of viral replication. You still have 20% of your normal virus load being released. Your person is still going to get sick. And those 20% of viruses that are escaping inhibition could lead to antiviral resistance. All of these things combined, the specificity needed for the drug, the use of animal models, the use of testing, makes drug discovery for antivirals in particular very, very expensive. So we've got to make it through a lot of hurdles. And right now you hear things about some researcher claiming that they're going to have a brand new novel coronavirus drug ready for use in 2 weeks. They're fast tracking things. The normal drug discovery looks something like this. We start out with a group that synthesizes or purifies, let's say, 100,000 different compounds. So that's the first step. Just make a bunch of compounds. Then we have to show that those compounds have an antiviral effect in cells. That if we treat cells in culture with these antivirals, we're going to get less virus or no virus released from the cells. And right away, a big chunk of them are going to get crossed off because they don't do that. So now we're left with like 20,000 of our initial 100,000. So we've already lost a ton. We next have to look and say, is this low toxicity in cells? Are we going to potentially kill the cells just by giving them the drug? And this is where we again lose a ton, right? Because the drug alone will be toxic to cells whether or not it has an antiviral effect. So we've gone from about 20,000 here down to now, look, we're at maybe like 300. With those remaining 300, we have to look and say, are these effective in animals? So maybe they're not toxic in animals, but maybe they don't get to the site of infection. If we're dealing with a hepatitis virus, maybe the drug never makes it to the liver. So we're going to lose a part here. We're down to about 100. Is there low toxicity in animals? Is the dose needed to have this antiviral effect going to have unacceptable side effects in animal studies? We lose a big chunk of these. Now we're down to about 20, right? We started with 100,000. Now we're down to 20. Once we make it past animals, we have to test for toxicity and side effects in humans, right? So what is fine in an animal may be different in a human. We're going to lose a whole bunch of them here. Now we're down to like 3 or 4 compounds left. And once we've passed the safety test in humans, we look and say, are these actually effective at preventing viral replication, at decreasing the virus load, at curing our humans? A lot of them won't be. And now we've gone from like our remaining 4 or 5 down to 1 compound that is going to get approved for use. This is all over the course of 10 to 15 years, not 2 to 3 weeks, not 5 to 7 months, over the course of years. And because you have to go through synthesis and all these different testing and phases of the clinical trial, it is not unusual for the cost required to bring this one antiviral drug to the market to exceed $100 to $200 million. So we can't simply just say, make new antiviral drugs and have that suddenly work. We need a lot of money for that. We need a lot of time. We need a lot of safety testing to make sure that this new drug that we put in humans isn't going to make our humans sick. Okay, so let's talk specifically about some antiviral agents. One common theme is to have inhibitors of those early steps of something like attachment. Some things that work pretty well is to use neutralizing antibodies. I don't know if you recall back in the 2015-2016 Ebola outbreak, there was a nurse in the United States who tested positive for Ebola, and she was actually given antibodies from a previous Ebola patient, the first doctor who came over to the US with it. And it seemed that those neutralizing antibodies actually helped her recover more quickly. So we can do something like that for a lot of other viruses as well, where we give a person antibodies to prevent the virus from attaching to the cell. We can also use peptides that will inhibit, or some sort of drug that will inhibit fusion of membranes, or that mimic attachment. So maybe they'll bind to the virus and they'll cover up those spikes that the virus would normally use to attach the cell, and now the virus can't attach. So these are really useful as preventative measures, but they're often limited in treatment. So by the time you already have an infection established in a person, these aren't going to do a whole lot to clear the infection. They may help some, like we think happened in the case of the Ebola nurse, but they're not great on their own. They make pretty good prophylactics though. The second is inhibitors of cell penetration and encoding. That's what Cora said she would target with the coronavirus. So we have several of these that are working against influenza, amantadine, which is one that you should know, and rimantadine. So both of these target the M2 ion channel, which prevents influenza uncoating. So if you remember, this is flu. It gets into the cell by endocytosis, and as that endosome starts to acidify, the M2 ion channel pumps that acid into the virus itself, which causes membrane fusion here and the release of viral genomes into the infected cell. So amantadine and rimantadine inhibit the M2 ion channel from bringing in the acid, so we don't get this fusion event and we don't get release and uncoating of the virus. So this is great, but it's really only useful against influenza A. It doesn't work against influenza B or C, and this year it was actually influenza B that was the more prevalent strain. So the other issue is that those human avian flu, like the bird flu that you hear about, are pretty resistant to these, so their use is pretty limited. Still talking about flu, another antiviral agent are the neuraminidase inhibitors. So if you recall, neuraminidase is required for release of influenza from the infected cell. Okay, so here we have our influenza virus. The red spikes are hemagglutinin, and the yellowy spikes are neuraminidase. So hemagglutinin attaches to sialic acid. Neuraminidase will cleave the sialic acid so our new viruses can be released and attached to new cells. When we use a neuraminidase inhibitor like zanamivir, you may have heard of it called Relenza, or oseltamivir, which you guys know of as Tamiflu, this actually binds to neuraminidase. So we can see that down here. These little black things would be our Tamiflu. They bind to neuraminidase and they prevent it from cleaving sialic acid. Okay, so now our newly made virus is stuck to the cell that it's trying to bud out of. It can't be released, so it cannot go on to infect a new cell. So we're not preventing any of the early stuff. We're still making lots of virus, but we're keeping it stuck to this one cell. These two, Relenza and Tamiflu, are effective against both influenza A and B. And I mean, they're effective, but they shorten the duration of illness by like a day and a half. And you also have to start taking them within, I believe it's the first 24 to 48 hours of symptom onset. Otherwise, they don't do anything to shorten the duration of illness. So they work, but again, they're limited in what they can treat. They're limited in how well they work and in the time frame in which you need to start taking them for them to be effective. The next big group, and there are a lot of these for different types of viruses, are the nucleic acid synthesis inhibitors. So in our yellow box here, we have our actual bases. So adenosine, guanosine, thymidine, and cytosine. And the nucleic acid synthesis inhibitors are derivatives of the normal bases. So they look in part like a normal base. They can be incorporated into replicating viral DNA or RNA, but then they affect what happens downstream. So we're going to talk about acyclovir here. It has a close relative, ganciclovir. But if we look just at the structure, right, so this looks like guanosine. It's got this whole same part up here, which is the actual, like, the nucleoside. And that structure is all the same. But we're missing our ribose with our free 3' OH, right? That whole chunk is missing right here. And there are others. AZT over here, you can see it is very similar to our thymidine. It has this same structure. It does have a ribose, but instead of the 3' OH, it has an ammonia instead. Okay, so some of the same things with minor changes that are going to affect the ability of a polymerase to add another base after these initial ones. Excuse me. So we have some that inhibit viral nucleic acid or DNA synthesis. These tend to be really specific against viral polymerases or reverse transcriptases. Okay, so these are ones that viral polymerases will incorporate much more regularly than DNA polymerases, or than host DNA polymerases. So it allows us to have some selective toxicity. So only viral polymerases will incorporate these into viral genomes. Host polymerases won't incorporate them into host genomes. If they weren't specific, if any old DNA polymerase could incorporate these, they would not be safe for use in humans. Those that target reverse transcriptases are safe because we don't really carry out reverse transcription because we're human. So acyclovir is one that's used to treat herpes viruses, particularly the herpes simplex viruses. You may have heard of ganciclovir. It treats cytomegalovirus, or valacyclovir, which is also used to treat both herpes simplex and cytomegalovirus. So acyclovir, this is it right here, is what we call a prodrug. So it enters the body in this form, and then it must be further processed into its final active form that gets incorporated into the viral polymerase. So we have our initial form. It itself is not active, but this is part of why it can be selectively toxic. So if this gets into a cell that's not infected by herpes virus, nothing happens. It just stays like this. It probably gets degraded. Nothing happens to the cell. And a cell that's infected with herpes simplex, that virus makes a little enzyme called tyrosine kinase, which is going to add a phosphate here. So this is the first step that has to happen. No herpes virus in the cell, this doesn't happen. After herpes tyrosine kinase adds that first phosphate, cellular kinases are going to add 2 more phosphates to it. So now it really looks like a nucleoside. This whole structure then can get incorporated into the growing strand of our viral DNA. You see it looks a lot like guanosine, again, only it lacks our ribose and our 3' OH. So in our growing strand of viral DNA, this gets incorporated, but because it lacks this OH, a new base cannot be added. So this strand of newly synthesized DNA is going to end here. We cannot further make viral genome, so we don't get complete genome replication. Pretty handy. And then this whole structure of acyclovir is pretty specific for viral DNA polymerases, and ours, again, don't incorporate it, so this doesn't affect our genome replication. Another nucleoside analog that inhibits viral RNA synthesis, and it also inhibits viral DNA synthesis, although we don't really understand how it inhibits viral DNA synthesis very well, is ribavirin. So this is another one you need to know about. There are several modes of action for this one. The one we best understand is what's shown here. I'll talk you through it. And this one gets used for the treatment of respiratory syncytial virus and hepatitis C viruses. So ribavirin, again, it's a nucleoside analog, so it looks like our bases. And as our viral polymerase copies and makes a new strand of viral RNA, it can incorporate our ribavirin in there. So ribavirin looks like, let's see, it looks like guanine and adenosine. So it can be incorporated in place of either of those. So here we see it's been incorporated into 2. We get synthesis of this whole strand. And then when we get a new plus strand being made here, maybe we have some incorporation of other ribavirins, but this is where it gets tricky. The polymerase doesn't know what to put across from, what to base pair with, that incorporated ribavirin. So it's going to put, right, we've got a C or a U here, we've got a new one incorporated. And as we keep replicating and making more genomes, now we're going to start to see some mutations occurring. So here in A, right, this is where we got our initial ribavirin, and it was base paired to a cytosine. Now we're getting a uracil put there instead. And over here where we had a uracil, right, we're getting a C put in place. And now we've got a U where we had a C. So we're accumulating a lot of mutations here as we continue to replicate our viral genome, which will result in just a bunch of non-functional genomes. So these will eventually not be able to make functional proteins, and we won't have new virus being released because the genome's just all messed up and we're not coding for any proteins anymore. As you can imagine, that can take a while for enough mutations to accumulate that nothing happens. So this is not a quick, you take it for a couple of days and you're better kind of treatment. And again, on this same line of having a nucleoside or a nucleotide analog that prevents replication of the viral genome, we have one of the most famous ones, AZT. So AZT is specific to inhibit reverse transcriptase in our HIV. So we do also have proteases that will target HIV, and we'll discuss those, and we'll discuss other types of HIV-specific drugs when we actually talk about that particular virus. But AZT, one of the most famous drugs, it looks like this. Okay, so it has this weird structure to it. Again, it has a ribose, but it has an ammonia instead of a 3' OH. Like with some of our other prodrugs, this has to be converted from this original to a drug that has our 3 phosphates there on the end. Once that happens, this whole thing gets incorporated into our DNA molecule that's being reverse transcribed in HIV. And again, because we don't have a 3' OH, synthesis of that DNA strand stops. So for HIV, that means we have our original RNA template. We don't get reverse transcription to DNA, so we don't get messenger RNA, we don't get protein, we don't get new virus being made. And kind of the last thing here on inhibiting biosynthesis would be the use of interferons. So we know that interferons can inhibit viral protein synthesis. They can also inhibit messenger RNA synthesis. So we can treat people with interferon alpha. That's been pretty effective in hepatitis B and hepatitis C treatment. But again, this is going to be a long treatment period. So for these viruses, it can be anywhere from 6 to 12 months of treatment. It takes a while because, again, we can't give people ridiculously high doses of interferon. That has some pretty bad side effects. So we have to give them a lower level for a very long time to control those viral loads and allow the body to clear the virus from the system. All right. So back to the beginning with our coronavirus replication cycle, we could, in theory, develop and find drugs that target any of these. Attachment, maybe we use some sort of receptor agonist that prevents this interaction from occurring. Maybe we use something that binds to those spike proteins and prevents them from binding to receptors at all. Maybe we prevent escape from the endosome in whatever mechanism that happens. Maybe we'll find that we can treat with interferon and that will inhibit all of this sort of stuff from happening, or maybe we'll find a way to prevent assembly. One of the other things that is being kind of tested right now, much like the use of the malaria drugs, it's very early phase. There's not been rigorous clinical trials of any appreciable number of people, but we're finding that drugs that prevent protease activity that are used for HIV and for hepatitis C actually show some efficacy against coronavirus. So In this step here, our genome gets into the cell. It gets translated into these big polyproteins, meaning a lot of proteins linked together. That polyprotein's non-functional. It's too huge. Imagine that you had 5 different proteins with different functions all linked together. As that one big protein, they can't work. We have to chop them up into their individual pieces. So that's the proteolysis step, and now we have our individual pieces that replicate the genome. We're finding that using drugs that prevent this step from happening appear to prevent the rest of this from happening. So some HIV drugs prevent various cellular proteases. And that seems to be working here as well. And the same for hepatitis C. Hepatitis C requires proteolysis of polyproteins into their subunits, and those seem to be working here too. Who knows, maybe in another 6 months to a year, there will be rigorous clinical trials, and we will see that there are actually some drugs to treat this virus, which could still be useful now, depending on where we are 6 months to a year from now, but would definitely be useful in the future if this virus or another coronavirus emerges, and we need to fight it off as well. Okay, so the last thing, antiviral drug resistance. Now, viruses, because they're so different from each other, and also because they're so structurally simple, they don't have any sort of horizontal gene transfer. So all of their resistance is based in mutation of the viral genome. Okay? So do we get minor mutational changes that are going to allow it to be drug resistant? So this would be the genetic drift that we talked about when we talked about influenza. How fast a virus mutates depends on a lot of different things. One is just the rate of replication. So the higher the rate of replication, the faster a virus replicates, is generally associated with a higher number of spontaneous mutations. So those that replicate really quickly are going to mutate really quickly. Another is, of course, selective drug pressure, same as what we see with antibiotics. So if we are treating with a drug, we're increasing the probability of mutations, or of the selection for mutations that are effective. So we're putting that bottleneck effect into place. And while those that are still susceptible to the drug are not going to replicate, those mutations that allow for escape from the drug are going to increase and grow. One way to get around this is combination therapy, like we see in the treatment of HIV, where we're not giving a person just one type of drug. We may give them 2 or 3 different types of drugs that target different parts of viral replication. And then the third is just the rate of mutation. So different viruses mutate at different rates, not just replicate at different rates, but accumulate mutations at different rates. So RNA viruses are more error prone. Their polymerases do not proofread. So those RNA dependent RNA polymerases don't have any sort of proofreading activity. So their mutation rate is about 1 in every 1,000 to every 10,000 nucleotides added. Right? So that's pretty regular. Imagine that this virus now replicates really, really quickly. And the number of new genomes you're going to get out from a rapidly mutating, rapidly replicating virus is going to be pretty high. So this alone will give us about 1 mutation in every 1 to 10 genomes made. So if you're making 1 million new genome copies in your cell, a lot of mutations are occurring. DNA viruses tend to either use our DNA polymerases, which are really good. They can proofread. They're what we call high fidelity. They don't make a lot of errors anyway. Or if they encode their own viral DNA polymerase, those are like ours, have proofreading capabilities, don't make a lot of errors. So we don't see as many mutations occur in our DNA viruses, or at least they don't occur as quickly in DNA viruses as they do in our RNA viruses. And one thing that's really interesting is that we can see when we look over time at the evolution of viruses through mutation that the mutation rate differs based on what viral gene we're looking at. So some genes are more susceptible to mutation than others. Because, think of it this way, if you get a mutation in a polymerase that makes the polymerase non-functional, you're not getting any viruses with that particular mutation coming out of your cell. So things like polymerases are not going to have a lot of errors in them because those viruses don't survive to make new viruses. But we can see mutations in things like the spike proteins or in things like matrix proteins that allow for assembly. So some genes will mutate a lot more than other genes will. All right, that is my last slide, guys. You've probably heard while we're talking about mutation that HIV is the fastest mutating virus we know of. It's actually the fastest mutating organism that we know of. And we'll talk a little bit about why it mutates so quickly later. It is an RNA virus, so it mutates pretty quickly, but a big part of it has to do with how terrible its polymerase is. Reverse transcriptase is terribly error prone. Which makes it mutate really, really quickly. All right, so those are our antivirals. Those are the basics of how they work and the ones that you need to know. And the reason for resistance we see here is mostly just mutation. Okay? All right, guys. That's all I have for you today. We'll come back next week and start talking about specific viruses and the diseases they cause.